Hi, everybody. I'm Wyatt. I'm a research plant pathologist for the USDA in Fargo. And as mentioned, I'm going to talk to you a little bit about CLS epidemiology and some of the work we're doing to track and model the fungicide resistance within commercial fields and how it changes and develops over time. A brief introduction to myself. I'm from Farmington, Minnesota, which is actually not too far away from here. I got both my degrees at North Dakota State University and got my PhD working on host pathogen interactions for barley and net blotch. And since August of 2021, I've been the research plant pathologist in the sugar beet unit, focusing mostly on Cercospora peticula and Cercospora leaf spot. We also do a little bit of work on some fusarium and are looking at other pathogens as well. So the core focuses of what my lab works on are Cercospora peticula's life cycle and CLS epidemiology. And we're interested in the life cycle because the better we understand the events surrounding things like sporulation, or when those spores land on a leaf, the germination, those environmental conditions and that process and that timing, the better we can then move over to the epidemiology side of things and start predicting infection, symptom development, spread of the disease, and so on. So these things basically flow into each other in a cycle. Now, before I dive into the results and the research I'm about to show today, I want to acknowledge that this is a huge collaborative effort. I started in 2021, and long before I got to the USDA, Gary Secor was working at NDSU and was collecting Cercospora isolates and phenotyping them for fungicide sensitivity, all the way back to 1997 is what we have data. And he's done that almost every single year for the last 20 some odd years. We have this very long temporal idea of fungicide resistance in the Red River Valley. And then my research leader in the sugar beet unit, Dr. Melvin Bolton, has done a ton of work looking at Cercospora's virulence mechanisms, the molecular biology of fungicide resistance and virulence, and all of that. So they've generated a ton of data that they've been able to just feed to me to look at when I start examining life cycle events, epidemiology, and specifically adaptation to our management practices. So the main issue we face right now in controlling Cercospora is fungicide resistance. And this is a chart of end of the year fungicide resistance testing from 2020, 2021, and 2022. And as you can see so on the left side of this graph is prevalence. Every single sample or every single fungicide has resistance at least over 50% over the last 3 years. And in 2022, red arrows here designate fungicide resistance prevalence at over 90%. Yellow designates under 80% fungicide resistance, but as you can see, this is all still very high. And this is to all of the fungicides that Gary Secor's lab tests for. So that's tin, headline. So tin, headline, which would be the QOI resistance, and then the DMIs, eminent, proline, inspire, and probosol. And this overall trend of increasing fungicide resistance over the last few years is a concerning thing, which is why we're starting to look into it more closely. Now, how this fungicide testing is typically done in Gary's lab is so leaf samples are submitted to his lab. They isolate out individual isolates. They plate those isolates out on these serially diluted fungicide amended plates, measure the growth on the unamended plate, and then they measure the growth on the fungicide plates to see at what concentration do we eventually see a reduction of growth to half of what the unamended plate is. And then we develop these curves and predict EC50 values from those curves, and then we can track that over time. And the main thing I want to get out of this slide here is that this has to be done independently for each fungicide tested and then each isolate. And so, for example, in 2022, 135 of the, I think, almost 600 samples that they did in Minnesota, and each isolate has to have 5 plates for each of the 5 fungicides they tested, and this turns into a pretty large number of samples to keep track. And this work is largely done by Viviana in Gary's lab, and it's a ton of work. which is why we can really only do it at the end of the season. We just don't have the manpower to continuously do this throughout the year. So one of the ways to get around that is with molecular markers. And so how we get to molecular markers is by understanding the mutations that have led to resistance. And previously, before I got here, we had these molecular markers developed for QOI resistance and benzimidazole resistance, so topsin. Melvin's lab and Gary's lab did a wonderful job of identifying the targets of these fungicides and then identifying the specific mutation, so the change in the Cercospora DNA that then leads to resistance. And one of the useful things about the QOI and benzimidazole resistance systems is that both traits are almost binary in their resistance and sensitivity. If you have the mutation for resistance, you are resistant. There's no in between. There's no gray area here. It's black and white, sensitive or resistant. And the same thing is true of the topsin resistance. If you have the mutation for resistance, you are resistant, and if you don't, you are sensitive. So this is a really nice system to set up and validate these molecular tests. But for the DMIs, the resistance is very much quantitative. And so you have a spectrum of resistance that's been associated with a number of different mutations, and we label those E170, L144F, and so on. But the critical part of identifying a molecular probe for DMI resistance would be to figure out what mutation leads to this first step. What's the critical mutation that leads to an isolate's ability to be resistant? And that's what we started digging at here. So Melvin's lab had already kind of isolated out two of these markers. This E170 and L144F is what they're titled, and they're both in the target for DMI. The target for DMI fungicides. And what you can see in these charts here is that if you have this GAG as opposed to this GAA mutation for this E170 mutation, if you have the A here, you get much more resistant isolates trending up. And if you have the G, they cluster down at the bottom, which would be sensitivity. Same thing can be said of this L144 mutation. If you have this G or this T, you get a clustering at the bottom here, indicating sensitivity. And if you have the C mutation, you start to see resistance. And so we wanted to see how well we could use these markers to predict resistance in some data we already had. So using the almost 600 isolates collected in 2021, which we had phenotyping data for already. So we already had the plate assays for this. We ran this set of markers to see if we could reliably predict the results from the plate assay. And the important part to get from this giant decision matrix here is that we were able to predict resistance to DMIs at a greater than 96% accuracy, which indicates to me that this is something that we can deploy readily as a rapid assay for DMI resistance. Now, one of the interesting things we noticed when working with this data is that not all DMI resistance is the same. And that was very evident by the fact that we saw that cross resistance between the four DMIs we tested, Proline, Eminent, Inspire, and Probosol, is not the same. And so this is the first line of evidence we have that there might actually be two classes of DMIs, which we've been arbitrarily calling DMI1 and DMI2, with DMI1 being containing tetraconazole and prothioconazole, which would be Proline and Eminent, and difenoconazole and methylfluconazole being DMI2. And what you're seeing in this chart here are the same isolates graphed with their relative resistance. So higher on this chart is more resistant. At the bottom, we have sensitivity. And what we see is that the same isolates are resistant to tetraconazole and prothioconazole, so Eminent and Proline, but they're sensitive still to difenoconazole and methylfluconazole, which would be Inspire and Probosol. And overall, the cross resistance between, for instance, Eminent and Proline, they correlate well, and we see 98% cross resistance. And for Inspire and Probosol, they correlate well, and we have 97% cross resistance. But when we compare the cross resistance between this group and this group, we see that there's a 98% cross resistance from Eminent and Proline to Inspire and Probosol, but the opposite is true of inspired probe salts and proline and eminent with 67%. So a third of the isolates we tested are resistant to proline and eminent, but not to inspired probe salt. So what this tells us in a roundabout way is that we have two different DMI classes here, eminent and proline, inspired and proposal, and our rapid assay is 96% accurate at distinguishing between resistance to both and sensitivity to both. So we can very quickly make a call based on submitted samples on whether or not it would be resistant here and sensitive here, or resistant to both. And so now, understanding that we have this E170 is the initial mutation that needs to happen, and we have a modifier in L144F, we were able to develop this and validate with this rapid test. And then that gives us a full set of molecular tools now to interrogate different parts of the growing season. Now, what you see in here is my conceptual understanding of the CLS disease cycle, kind of broken it up into pre-infection, which would be, when the primary inoculum is exposed, so snow gets off the field, dirt starts kicking around, those spores start getting in the air. Those spores will disperse, and then at some point we get latent infection. And typically we see this at the 3 to 4 leaf growth stages. And before that, we don't detect any latent infection. And then sometime between latent infection and identifying first spots in the field, there's a whole black box that we don't quite understand. Once symptoms develop, we have new spores, and new spores then kick off this cycle of approximately 14 days with spores dispersing, new infection occurring, latent infection, more symptoms, more spores, and the cycle goes on until we harvest beets. So these molecular markers that we have now give us an opportunity to take a peek into these different time points along the disease cycle to see how fungicide resistance is developing and changing, and also specifically with latent infection to really pinpoint the start of infection in the year. So how this works for detection, we're really reliably able to detect Cercospora DNA in asymptomatic leaf tissues by using our molecular marker for QOI resistance. And the reason for that is the target of QOI fungicides is a multi-copy gene. So if you can imagine, if we're able to really reliably detect a single copy of something, having two would be 2 times better. And so this is a very sensitive and very specific assay that we're able to run. And the output looks a little bit like this, where we have these fluorescent curves, and a positive curve is anything that passes our set threshold, and a negative curve indicates that there's no CLS. So using this assay, we're able to detect Cercospora in asymptomatic leaf tissue. Now, when we ran this on a wide collection of samples from 2022 Red River Valley wide, we saw that the prevalence of CLS in submitted leaf samples rose steadily from the middle of June until the first week of July, at which point we had 100% of our samples having CLS detected growing asymptomatically in the leaf tissue. That trend, when we break the Red River Valley data into just the southern mini growing region, we see the same thing. And one of the interesting points of the chart you're looking at here is that as far as asymptomatic infection is concerned, we don't really see a difference between CR plus and non-CR plus varieties, with CR plus varieties being shown here in red and the blue dashed line indicating non-CR plus varieties. Now, this is our first peek into latent infection, and this gives us a really useful data point to start modeling for disease risk, because now we know the fungus is there at least by the first week of July, and this lets us start looking at environmental conditions in the preceding weeks that might be indicative of the start of this latent infection. And it also gives us a really useful time point for the start of latent infection and that time span between latent infection and first spots in the field. So now we can start to really focus on knowing that we have infection, and then what environmental conditions are actually predictive of disease lesions in the field. Now, as I mentioned before, we have molecular assays for all 3 of these fungicides. And so we were really interested in seeing how that fungicide resistance developed at the beginning of the year. So we ran all 3 of those markers on all of those samples. And what I'm going to show you right now is QOI resistance prevalence and toxin resistance prevalence as we get into the actual infection stage of the season. And the important part on all of these charts to focus on is really this last third of the chart, which would be when prevalence of latent infection was 100% in all of the samples that we had submitted. And what you can see here is that as infection prevalence rose, we got a pretty stable indicator that QOI resistance resistance prevalence hovered around 35%. And that our toxin resistance was skyrocketing to greater than 90%. So going into the season, generally speaking, we have a fair amount of QOI resistance and a ton of toxin resistance. Now for the 2 DMIs, noticed opposing trends. So in eminent, we had rising resistance going into the actual saturated infection stage of the season. And for Inspire and Propofol, that result was just the opposite. We had almost no resistance. So only 4% of our samples showed resistance to Inspire and Propofol, as opposed to about 80% showing resistance to Proline and Eminent. And one of the main findings of this, or the main takeaways, is that if you look at our end of the year testing, shown in gray from 2021, our latent infection testing, shown in yellow for 2022, and then our end of the year testing for fungicide resistance in 2022, we see some discrepancies between what happens at the end of the year and then what actually starts as infected material throughout the year. But the other interesting part is that we do have some consistency in the end of the year testing, meaning that our fungicide application management practices are consistently producing the same levels of resistance year to year at the end of the year. For Eminent and Proline, it seems like that resistance is pretty pervasive and ever present. But again, the interesting part is the reduction of resistance to headline at the beginning of the year and the reduction of resistance to Inspire and Propofol at the beginning of the year. Real quick, I just want to touch on CR Plus and its impact on fungicide resistance. So for all the samples that were submitted from 2021 at the end of the year, our latent infection samples from 2022 and 2022 end of the year data, we see this similar trend where specifically for the triazoles, for the DMIs, we see a reduced level of resistance in CR Plus derived isolates. And so that's shown on these 4 charts here for Eminent, Proline, Inspire, and Propofol. Yellow is CR Plus varieties, blue is non-CR Plus varieties, and any higher shift on these charts would indicate more resistance, and clustering towards the bottom would indicate lower resistance. And this was actually statistically significant for Proline and Propofol, and nearly significant for Eminent and Inspire. But as I layer on 2022's data, I would anticipate we'd start to see some real discrepancies. So CR Plus derived isolates appear to be more sensitive to fungicides, and right now our hypothesis moving forward is that that's actually due to being sprayed less, and not necessarily due to anything that the CR Plus is imparting, but its resistance allows us to spray that less, and so we see less resistance develop over time. Now, another brief focus on CR Plus here. Of those isolates collected off of CR Plus, we did whole genome sequencing on them, essentially to get genetic fingerprints that we could then check to see if there's overlap between isolates coming off of CR Plus and isolates that are coming off of non-CR Plus material. And the overall result of that is that if you were to think of the population that's present on non-CR Plus material, the population on CR Plus is similar, but reduced in its diversity. So my small cartoon here is just showing that essentially the CR Plus is filtering out isolates that can't infect it, which would be indicated by the blue stars here that can't pass through this small channel, which we describe often as a bottleneck. And then isolates that can infect CR Plus do. And from here, what we would anticipate happening is further optimization on CR Plus, because if they can infect it, they will find a way to infect it better in subsequent seasons. And all of this to say, CR Plus is a great tool, but if we don't also manage this properly with fungicide applications, you will eventually see adaptation, and then this useful tool will become far less useful. So jumping back into the disease cycle and timeline, the other opportunity we have for tracking fungicide resistance is actually during the season now. Because of the rapid nature of these assays and the high throughput nature, we kicked off a small pilot study looking at 3 fields where we collected 84 leaf samples spread across the diagonal of the field. And we did this collection before any fungicides were applied. Then after the first application, but as close to the second application as possible, trying to give the pathogen population the maximum amount of time to recover. And then we did that for every single application, and then a collection finally at the end of the season. And so for these 3 fields that were located fairly close to Fargo, North Dakota, because I had to make the drive and do the collections, we collected 5 times throughout the year, and there were a total of 3 fungicide applications applied. Sorry, 4 fungicide applications. So the main finding, or a primary finding of this pilot study, is that prevalence rose to about 100% and then never dropped. So no matter how many fungicide applications were applied, we never saw a decrease in the prevalence of infected tissue for the 84 samples per field. However, we did see some really interesting trends as far as the fungicide resistance that we saw in these fields. So when looking at QOI resistance in this field actually started fairly high. However, following the first DMI and EVDC application, we saw QOI resistance plummet to what is about 6%, and then steadily rise to about 11%, right up until a QOI was applied at the end of the year, and then we see the spike back to what we started at. So this is obviously preliminary data. This is 3 fields from a single year, but this may indicate that QOI resistance is fairly fragile. And following a application with a DMI or a tin topsin, I should say tin or topsin, it's the specific combination that leads to this. You see a rapid reduction in that resistance level, and so this may indicate that this could be a useful tool during the season as well. When we focus on the DMI, so DMI 1 here, which would be eminent and proline, after this first DMI application, we see resistance to proline and eminent skyrocket to almost 100%, and then it maintains itself at this very high level, which is interesting because you would anticipate that when you apply a tin topsin later in the season that you would get a reduction, but we don't see that. And I guess if you were to move into this next part naively, I would have assumed to see the same trend in resistance for DMI2, which would be Inspire and Provosol. But we actually don't. After the first DMI application, which was actually Inspire in this case, we see a slight increase in the resistance to Inspire and Provosol, but then a reduction following the tin topsin that followed this up, and even a further reduction following the later DMI application. And so this dynamic is very interesting to us because it may indicate that there's a best order to applying these DMIs. Especially given the lack of the decreased cross resistance between DMI2 and DMI1. Couple takeaways to wrap this up. We now have molecular assays for QOI, DMI, and benzimidazole resistance. And we're able to use those to rapidly and efficiently test asymptomatic tissue as well as infected tissue throughout the year to determine fungicide resistance profiles for these 3 very important fungicides. We were also able to establish that there's 2 categories in DMIs with different cross resistance. Eminent proline grouped together, Inspire Provosol grouped together. And these molecular assays really are very useful for rapid testing throughout the year. We also were able to get our first snapshot of the start of CLS infection with this latent infection study and get an idea of what our fungicide resistance profiles look like moving into the season, which would be useful, especially if you, let's say, detect a ton of resistance right away in latent infection samples or resistance to proline. It's a good idea to not spray proline then right immediately following that. And gaining a better understanding of how fungicide resistance dynamics change throughout the growing season, especially in response to other fungicide chemistries being deployed. So knowing that QOI resistance, for instance, has a tendency to decrease following a DMI application may be a useful tool for us to push the pathogen population in a direction that we want to set up the next fungicide application. CR plus right now is likely contributing to reduced fungicide resistance. And we think that's likely through decreased application of fungicides, but yeah, that's again, one and 2 years of data. So we'll wait to see what the actual cause of that is. And then generally speaking, the population of Cercospora infecting CR plus is really just a reduced representation of what's actually in the overall population. We refer to this as a bottleneck, but a better analogy might be that if you go to like the state fair and they have those lines that say must be this tall to ride, that's what CR plus is doing to our Cercospora population. Moving forward, we're going to continue our latent infection assays. We're expanding our in-season epidemiology to track these fungicide resistance dynamics. So we're going to do a much larger study in 2023 instead of just 3 fields. And we're going to work to identify the underlying genetics for TIM resistance, because right now that's the most important fungicide that we don't have a molecular assay for right now to detect resistance. And we're going to continue sampling Cercospora from CR plus sugar beets to monitor for continued adaptation. I have a ton of thank yous to make. I'll make this quick. Melvin's lab has been instrumental in all of this research, as is Gary's Seafor's lab in plant pathology at NDSU. And then everyone who was involved in the latent infection study this year across the Red River Valley. I mean, could not have done that without any of these people, and specifically the agricultural staff. With that, I can take any questions you might have.